Hello everyone, welcome back another class of regeneration biology. In today's class, we will be learning more in detail about the different interactions the cells have on the scaffold, and to some extent we have seen it in the previous class, that is adhesion, migration and aggregation of stem cells. We will revisit very quickly and we'll move on to its implications in organ culture. So if you look an overview, the stem cell adhesion, its migration and aggregation are crucial for their development, function and therapeutic applications. In yesterday's or previous class, we have seen how the adhesion is measured, how their interaction with the environment is determined, and how you evaluate the migration and aggregation and settling down efficiency for creating an environment in which the cells are able to form an organoid. So in today's class, we will learn how this has been put into reality and put into practice. So the adhesion allows the cells to attach to other cells and also to the extracellular matrix, and this facilitates the interconnections between the cells and also favor the signaling, cellular signaling events. So the migration involves the stem cells moving throughout the body, which occurs in a real-time scenario, and it is often guided by various signals to reach specific locations. Mainly they are called chemokine signaling, where the cells will migrate from location A to location B. And this is very prevalent in locations like bone marrow or injured tissues. The dynamics or the movement of the cells are very high in these tissues. Aggregation, on the other hand, refers to stem cells assembling into three-dimensional structures which can influence their biological properties and behavior. So if a cell is not able to form aggregate, then it is less likely a suitable cell type or a isolate for an organoid or organ culture. So adhesion, let us quickly visit. Cell-ECM interaction is often referred to as adhesion. Stem cells are specialized adhesion molecules like integrins and selectins, which is present on the cells and bind to the components of the ECM. That's why the ECM is different in different tissue types. Although molecular level they will be more or less the same, but there will be some unique proteins present in individual cell type. Similarly, if you are using an artificial material as a scaffold for organoid culture, we need to ensure the cells which you are planning to culture has got a given set of interacting proteins like integrins and selectins. So my scaffold should allow interaction with those molecules. And ECM can have specific proteins such as fibronectin, collagen and laminin. These are some of the ECM proteins. And cell-cell interaction, that is also part of adhesion. Adhesion molecules also mediate cell-cell interactions, allowing stem cells to interact with other cells and form tissues. So this interaction is a must, without which no cell is going to multiply. Then comes the signaling event. Just interaction is not good enough, signaling should kickstart. Adhesion events can trigger intracellular signaling pathways affecting stem cell survival, proliferation and differentiation itself. So what is the importance of all this? In a nutshell, you can say proper adhesion is essential for stem cell survival, localization and function in different tissues and different environments. So now look into the next aspect, that is the migration. So migration has got one unique feature that is called homing. Homing basically means the stem cells, particularly hematopoietic stem cells, actively migrate to specific locations, a process known as homing, often guided by chemokines and other signaling molecules, without which a given stem cell will not be able to distinguish themselves from the rest of the stem cell. They often need to distinguish themselves simply because not that every stem cell is going to give rise to only one type of cell. Say in bone marrow, we know we have myeloid lineage and lymphoid lineage, but they are coming from the same parent cell. So who tells one cell to become myeloid lineage? Who tells the same cell to become a lymphoid lineage? So this kind of variations are possible because of their homing behavior. And mechanotransduction. Stem cells can sense and respond to physical and mechanical environmental cues influencing the migration. Say a stem cell is taken and you bend it. So it is stretching the cytoskeletal and ECM interaction in a given way that itself can trigger a proliferative response. So the mechanotransduction is very, very important for the behavior of the stem cell. Then comes the chemotaxis. Chemokines like SDF1, one of the chemokine, attract stem cells towards specific locations such as bone marrow or to the injury sites. And what is their importance is the migration allow stem cells to reach tissues which needed repair because that is damaged tissue. If you have an injury, of course there is a damage. So you need cells. So if no cells are coming there, naturally that wound will never get healed. And also this allows these chemokines are specific for wounded tissues or a cell undergoing apoptosis, and this will allow the stem cells to reach tissues that need repair, regeneration or development. So now let us see the aggregation. Three-dimensional structures are basically resulted from cellular aggregation. Stem cells can self-assemble into 3D aggregates. 3D means three-dimensional aggregates, mimicking a tissue architecture and enhancing their biological properties. A tissue can perform its task not when it is in 2D, two-dimensional. Often it needs a three-dimensional structure for performing the task optimally. So influence on its function. So aggregation can affect stem cell proliferation, differentiation and secretion of growth factors, potentially improving their therapeutic efficiency. So that is why aggregation is so important, because it can influence the function of the stem cell itself. And the techniques used are different methods can induce stem cell aggregation, including gentle stirring or controlled detachment from solid surface, etc. Whenever you give a altered scenario or an altered environment to the stem cell, they tend to aggregate. So the importance of aggregation is it can improve stem cell delivery, retention and functional activation in vivo, leading to a better therapeutic outcome. In summary, adhesion, migration and aggregation are interconnected processes that play crucial roles in stem cell biology and therapeutic applications. So understanding these processes can help us in designing stem cell based strategies or therapeutic strategies and for various diseases and injuries. So that is why understanding these three pathways are very important: adhesion, migration and aggregation. So now let us see in an organ culture perspective. In an artificial organ culture, using stem cells for regeneration, adhesion and migration and aggregation are crucial for tissue formation and functionality. This we have already seen it and discussed. Stem cells must adhere to a substrate, migrate to the appropriate location. Why? Because then only it can signal to another cell, plus it can differentiate into a given trajectory and aggregate to form the functional tissue structures, and then it should be able to form aggregate so that the tissue can have a multi-cellular structure which has got uniqueness in each and every cell. Say if a tissue has got 1,000 cells, not all 1,000 are equal. There may be 10 different types of cell. On an average, there may be 100 individuals each. So 10 into 100,000. So this way the organ can perform better. So you don't want in an organ all cells are behaving or performing the same way. That is not how organs are designed in a body of an organism. Engineering these processes in a controlled manner is key to creating functional artificial organs. So how these processes are controlled? Let us see. This is influenced by different conditions. One is biomaterial properties. What scaffold you have used for making these culture, stem cell culture environment. The type of biomaterial used in the artificial organ culture, including its stiffness, topography, and chemical composition, can influence cell adhesion, migration, and aggregation. So the growth factors and signaling molecules also can have a big say in this controlling of these processes. The molecules can guide stem cell differentiation and migration within the artificial organ, ensuring the formation of desired tissue structures. So that is why the growth factors and signaling molecules are important. So three-dimensional culture. The culturing stem cells in a three-dimensional environment such as hydrogel or a spheroid can promote aggregation and mimic the natural tissue organization what is seen in vivo. So you don't want an organ that is unique. I made a unique kidney or a unique liver which is very different from what is seen in human. Of course, it's a good idea. However, that liver has to work in conjunction with rest of the organs present in our body. So hence it is always good that the liver you make or a kidney organoid you make has to be similar or as close to the real tissue as possible than making something in a shortcut or a unique manner. So some examples of its application. One is bone regeneration. Stem cells can regenerate bone tissue by promoting bone cell adhesion, migration, and differentiation within a bone-like scaffold. So you make a bone-like scaffold, first inoculate them with stem cell, allow them the appropriate guidance cues, then it will differentiate into bony plates and you will end up getting a bone. And cartilage regeneration. Stem cells can repair damaged cartilage by promoting chondrocyte proliferation and differentiation within a three-dimensional cartilage scaffold. So you can design based on what should be the ultimate organ which you are desiring or ultimate tissue what you are desiring. So organ regeneration. Stem cell based approaches are being explored to regenerate various organs including liver, heart, kidneys by inducing stem cell differentiation and migration within the biocompatible scaffold. So these are actively emerging area and some examples we will see how it is being implemented in a human patient. Let us see the 3D stem cell spheroid and organoid systems for tissue repair and regeneration. This picture is an overview. So 3D stem cell culture systems that mimic in vivo environment enable basic research about various tissues and organs. So the therapeutic applications of stem cell based spheroids and organoids, including engineering techniques and tissue repair and regeneration, has completely changed or revolutionized the regenerative medicine in total. So the three-dimensional stem cell culture systems have attracted considerable attention as a way to mimic better the complex interactions between individual cells and extracellular matrix that occur in vivo. That is what I told you already, that it should mimic exactly like how a natural organ or a natural organism is functioning, and those interactions should be brought in. Moreover, the 3D cell culture systems have very unique properties that help to guide specific functions, growth and processes of stem cells. For example, what is seen during embryogenesis, morphogenesis and organogenesis. So this has to be simulated. These embryonic properties or little bit later when you have morphogenesis or much later when you have organogenesis, these features should be copied into organ culture. As you can see here, you take stem cells and this is you can apply different forces to adhere and form organoid structure. One is a hanging drop and another approach is in a microwell. So you can use different matrix, different microfluidic approach, sometimes use magnetic force, and you can spin them, like churn the cells so that they will form aggregate. Once you have the spheroid that is formed, and that can automatically give rise to a stem cell organoid, which is ready for transplanting into the damaged organ or part into a patient. That is what this cartoon is depicting. So if you look at historical perspective, it's always important to remember the history where from we have come, and this picture is depicting exactly that. It started in 1907, 1907th year it has started, and we stand now 2025. So you can see what are the steps that has happened, and those who are interested can read this reference I have given here. A few of them are listed here. 3D cell culture has a history of about 100 years. In 1907, Wilson et al. Described the first attempt of reorganizing an organism by demonstrating the ability of dissociated sponge cells to self-organize into a whole organism. We have seen this in Hydra also. We call it as morphollaxis. Just to take 100 cells, put them in a petri dish, you will end up getting a miniature Hydra which will grow to the adult size. That same year, the hanging drop method was established by Ross and Harrison et al. While exploring ways to culture and maintain frog embryo nerve fibers in vitro. This is another approach done by Ross Harrison. Thomson et al. Later successfully isolated cultured human embryonic stem cells derived from human blastocysts in 1998. That was the beginning of the so-called stem cell era. Later, the Yamanaka group first established iPSCs by reprogramming mouse and human fibroblasts, significantly influencing stem cell and organoid research from 2006 to 2007. In 2008, Hiraku and colleagues generated self-organized and polarized 3D cerebral cortical tissue from mouse and human embryonic stem cell. That was a breakthrough experiment. In 2009, Sato et al. Reported that 3D intestinal organoids formed by adult mouse intestinal stem cells isolated from primary intestinal tissue. So, lot of research is going on. A few of them are listed here. A lot of epic research have gone through to make the regeneration biology the stage where what we are seeing right now. The future prospect, if you look in the near future, applying microengineering and nanoengineering based platforms such as particle fibers, structural cues, fluidic chips, and 3D bioprinting at the micro and nano scale may facilitate the increased production, improved reproducibility, and development of highly matured organoid system, which is very much likely that is going to happen. Let us quickly visit what are the 2D cultured stem cell are used, how they are applied. So far we discussed about 3D. Of course 3D is good in making organoids, etc. Let us quickly visit how beneficial or how useful the 2D culture. Transplantation of stem cells can be utilized for the treatment of diseases such as the regeneration of tissues and organs instead of using complex surgical procedure or tissue and organ transplantation. In recent decades, tissue engineered using a variety of stem cells have been applied to epithelial surfaces. Epithelial surfaces means you do not need the so-called organoid structure or organoid culture. You can simply have a two-dimensional structure such as skin, cornea, mucosal membranes, and hard tissues. And examples include bone, tooth, enamel, dentin, and cementum, etc. need only a partial repair. You don't need to have the so-called organoid culture. Stem cell-based therapeutic applications for tissue regeneration are done by transplanting only stem cells with controlled functions or engineered scaffolds combined with stem cells. So it's a combination of these two are done, stem cells and the scaffold, especially made for specific therapeutic application. various engineering approaches are used to improve adhesion, proliferation, differentiation, migration, and paracrine factor secretion from the stem cells used to regenerate various tissues such as bone, cartilage, tendon, muscle, nerve, tooth, skin, etc. Micro and nano engineered platforms and biomaterials have been utilized to modulate the behaviors of stem cells and that can enhance their function by providing them with specific microenvironment. So the 2D cultured stem cells also have got tremendous potential when it comes to regeneration biology. So let us see what are the approaches and tissues which are done using 2D approach examples. Bone tissue regeneration, cartilage tissue regeneration, tendon and muscle tissue regeneration, nerve tissue regeneration, dental tissue regeneration, skin tissue regeneration, they all can be manipulated more easily by 2D approach. So let us see what is the future of 2D culture when you want to use it in a therapeutic scenario. Currently, the most commonly proposed stem cell therapy in clinical trials is performing in vitro expansion of a sufficient number of autologous stem cells. That autologous means from your own body, isolated from the patient and then injecting them into the target site. That is a simplistic workflow. However, stem cells transplanted into the target site have critical limitations such as low efficiency. That is, cells can be easily washed out at the target sites due to various reasons, because they were not able to establish or they may not be able to adhere to the existing tissue, or the immune system attacked. Even though they are autologous, maybe some uneasiness or some strangeness may be felt from these stem cells by the immune cells. So, and the failure to maintain their viability and function, and because they have to self-renew also. Sometimes self-renewal may get affected. Multi-lineage differentiation, you wanted X tissue, now it is not making X tissue, it is making Y tissue, because that also the stem cell have the potential. So what you need, you may not get it. It's almost like if you are in a boat, you want to go in the next direction, you are most likely go with the flow of the water. If you don't have an oar or you don't have a stick to regulate the movement, then whatever direction you turn, the boat will go, or that float will go wherever direction it feels like. So this possibility is there if you put a stem cell in a defective organ. If it is a perfectly functioning organ, you don't have to put it. Defective organ, it's already underperforming, so it won't get the right cue for the stem cells to differentiate. So this has resulted in limited success in restoring damaged tissues and organs, especially in large scale tissue repair or regeneration. Therefore, effective delivery systems that regulate the stem cell survival, behavior and function are required to efficiently transplant stem cells into the target sites. Recently, many studies demonstrated that 3D cultured stem cells enhanced the viability, differentiation, paracrine secretion and tissue regeneration compared to 2D cultured stem cell. So 3D culture stem cells and spheroids and organoids. Cardiac stem cell spheroids promoted the secretion of growth factors and the expression of cardiomyocyte specific markers. The spheroids improved cell engraftment and survival within the myocardium and enhanced neovascularization and myocardial regeneration, resulting in myocardial infarction recovery compared to single cells. So therefore, the 3D cultured stem cells can be good alternatives to stem cell transplantation due to more realistic biochemical and biomechanical microenvironments than the 2D cultured stem cells. So the spheroids formed by aggregating stem cells result from the self-assembly behaviors of single cells in suspension due to the embryogenesis, morphogenesis and organogenesis, which we discussed already. The formation of spheroids involves complex, homogeneous and heterogeneous binding of the cell adhesion molecules, ECM proteins and integrins. Stem cell spheroid assembly occurs in multiple steps. First, the single cells are drawn closer to form loosely adhesive cell spheroids because ECM fibers and complementary binding of the peripheral cell surface to the integrins can encourage the preliminary aggregation. That is what we want. Next, the cadherin on the cell membrane surface induces tight connections between the aggregated cells because of the homophilic cadherin binding of the peripheral cells. So, cadherin can favor the aggregation in a nutshell. Finally, early cell assemblies formed by both pathways generate contractile forces via the rearrangement of actin stress fibers leading to the compression. Like I told you, the mechanotransduction lead compression and the formation of mature spheroids. So after these processes, strong, compact, multicellular spheroids are formed. Depending upon their complexity, 3D cell structures systems can be classified into three major organization and structural types: spheroids, multicellular spheroids, and organoids. Spheroids are generally considered 3D cell aggregates generated from single or various cell types but cannot completely mimic the complex contact of other cell types. Stem cell derived organoids have a similar phenotype to the in vivo situation, which has higher tissue complexity than spheroids. Although the differences between spheroids and organoids are vaguely defined as of now and consistent among the researchers, stem cell spheroids are usually close to the meaning of simple stem cells aggregates that can be utilized in a series of processes or developing organoids from the stem cells. So the internal developmental processes drive organoid formation. Spheroids develop primarily through cell-cell adhesion. Stem cell-based organoids can be formed by providing the appropriate physical and biochemical signal for differentiation and development. Although you made the organoid, it has to differentiate also eventually in the host. The organ-like phenotype or the embryoid body after forming stem cell-based aggregates and spheroids. So the engineering techniques for the formation of stem cell spheroids. So the generation of stem cell spheroids is based on the common self-assembly principle. Cell self-organization occurs within the test tube if the cells cannot attach to the substrate surface and thus must interact with each other. Naturally, you will get the spheroids. The tight connection, cell-cell communication, and Cell-ECM communication within the cell spheroids contribute to the controlling stem cell behavior and functions. This is the outcome of the spheroid formation, such as viability, stimuli responsiveness, protein secretion, etc., leading to considerable differences compared to the monolayer cell culture. Spheroids can perform differently. The high throughput nature of generating stem cell spheroids with uniform size and composition is an important factor in fabrication of the whole assembly unit. To maximize the functionality of stem cells and cell-cell ECM connections when culturing 3D spheroids, various platforms based on commonly used spheroid formation methods such as hang drop, well plate, and spinner flask, and hydrogel matrix, magnet, microfluid chip, etc. have been developed. So commonly used formation methods and advanced engineering formation methods of stem cell spheroids are listed here. You can see the different approaches are like a hang drop method, well plate method, etc. are listed here. And here you can see there is a magnetic particle, magnetic force application, etc. are done to create the spheroids. So we can understand the spheroids and organoids have been developed for advanced therapeutic application. Functional 3D stem cell culture adopted for tissue regeneration and repair, for state-of-art organ culture, and also the advanced engineering technologies have been applied day by day to improve the quality of the organoids. So we will learn more about the organoids and the spheroids in next class. Thank you.